I'm Gail.
And hi, I'm Catherine. Welcome to Women Over 70, Aging Reimagined, our award-winning weekly podcast. Please visit womenover70.com and consider joining Aging Reimagined Circle, our sustaining membership fund, so we may continue to inspire women to age with curiosity, courage, and creativity. Members enjoy monthly programming and probing discussions. We hope to see you soon on Zoom.
Coleen T. Murphy is professor of genomics and molecular biology at Princeton University. She's director of Princeton's Glenn Foundation for Research on Aging, director of the Lewis-Sigler Institute of Genomics at Princeton, and director of the Siemens Collaboration on Plasticity in the Aging Brain. Coleen's lab focuses on the process of aging, which remains one of the fundamental mysteries of biology. Her recently published book, How We Age, The Science of Longevity, which I have right here. It's a wonderful book. Recounts her extensive research of model systems, particularly simple invertebrate animals, combined with breakthroughs in genomic methods, which have allowed scientists to probe the molecular mechanisms of longevity and aging. While your book is scientific in nature, it's surprisingly readable, even by a layperson. You provide a guide to the science, what we know about aging, how we know what we know, and what we can do with this new knowledge. And you do it all with clarity and wit, Coleen. So welcome to Women Over 70 Aging Reimagined.
Thank you. Thanks for having me.
You're welcome. Now, when we spoke, you said to me that the real trick in science is knowing what the right questions are to ask and how far you can take it. So how did you know the right questions to ask, and how far do you hope to take this?
I mean, that's a great question. So that's the benefit of having done when you do a PhD, you learn how to do a lot of work and know your background really well. And I think that's part of it. It's easy to jump into some new field and assume that you'll automatically come up with really great questions. But often people have asked all the kind of low-hanging fruit kind of questions. And so you have to delve deeper. Mm-hmm. And so that's one thing, just knowing the background well enough to be able to ask some questions, but also then coming in from a different angle. So you don't ask the same question that other people have retread over and over again. And then I think the third part of that is the development of new tools that can allow you to ask questions that people haven't been able to, just because they haven't been able to, delve deeply enough into some technical side. And so one of the things that's really benefited us is the development of genomics, which allows us to look at all the changes that happen in an animal simultaneously instead of just trying to drill down on one thing. And that's given us a really unbiased view of, different phenomena that we've studied in aging. So that's a technical part of it. And the other part is, you can do a million experiments that don't really ask new questions or answer new questions. So really trying to think deeply about what is it that we don't know yet, and what would be the best way to answer that question is, I think it's time well spent if scientists spend a little bit of, especially students, thinking about that, because it's really easy to read literature in your field and then say, oh, that must be the interesting thing to do. When in fact, actually, there could be really interesting questions that no one has thought of, or they haven't developed the tools to answer them. So that's why they don't read about them. So you kind of have to dig around a little bit first.
So where are we actually in understanding aging, and what does it mean to all of us in terms of longevity? I mean, your book, if I might add, while it's very scientific, I find myself picking it up, looking at different aspects of it, just finding a page and finding something that's really of interest.
That makes me so happy to hear, because, I sat literally at this desk for several years, toiling away to write this. So I'm really happy that you're finding it both readable and interesting. So where are we at? Okay. So I think everybody knows that if you went on anywhere on the web right now and typed in something about aging, you get an answer, right? You'd go, Google would give you some answer. And often those answers are kind of the things that we already knew for many, many years. So I feel like an almost obsession with dietary restriction as something that will cure all ills. And, to some degree that is a little bit true. Like there's a lot of things that dietary restriction can benefit, but I'm not really a fan of dietary restriction for a lot of reasons, both scientific and basically personal. I don't really want to starve myself. And so, I'm very interested in trying to understand what are the things that we could do that don't necessarily require dietary restriction in particular. Are there other things that we could do that would help us preserve our health, our cognitive function, for women who are younger, reproductive function? Are there ways that we can do that don't rely on things that we already knew about for 30 years? And so that's what most of the aging field is about, which is different from what you'll read on the internet. Internet, part of the problems of writing this book is that, Google you get kind of all kinds of advertisements, and it constantly is, throwing intermittent fasting advertisements at me. I'm really that's not really what I'm interested in. There's so many different pathways that are involved in aging, and the coordination between different tissues and the molecules and the molecular functions. So back to your question, like, what do we really know now? We really understand. This is the work of thousands of people in different labs, many different molecular pathways, how they're regulated, what changes with age, how we can slow things down. And so what's happening now, it's actually becoming true. Like, maybe 10 or 15 years ago, it would have been weird to say, oh, we're going to find a pill that's going to do something good in the, aging realm. And actually, there's a lot of biotech companies that have taken that information that we've learned in academia and really moved it forward. And so we're starting to see the fruits of that research. And I think in about 10 years, we'll really have a lot of ways to, things that could be prescribed for different, phenotypes, different aspects of aging that would be very beneficial.
Yeah. And go ahead, Ken.
I just want to say that I so appreciated the emphasis that you gave in the book about it's not just longevity, it's living healthy for a longer period of time. And I think so many people seem to be afraid of aging, and then it might get in the way of them thinking about, well, how can I be healthy and live longer? And maybe you could speak to that a bit more.
Yeah, it's funny because when I started my lab back in 2005, this quality of life question, quality of life with age I was kind of obsessed with it for a different reason. That was like, I realized when I was a postdoc that even if I could live to be 150, if you give me a pill to live 150 years, I would still have to make all my life decisions about when to have kids within the next couple of years, right? Because nobody had worked on things like reproductive aging, which is another quality of life aspect. And, I think we're all concerned with that. We want to keep our brains working as long as possible. And so if, there's this weird idea that if you live longer, you're necessarily going to keep your brain working longer, which may be true or it may not be true. And so that's why it's really important to ask the question, how can we extend, brain function with age? And is that through the same pathway or different pathways that would allow the rest of our body to live long? And I think it's a complicated question because it's partially right in both directions, right? There are pathways that seem to, like we study the insulin signaling pathway, and in the model systems we've worked on, extending lifespan in those animals also extends their brain function, okay? But it's not necessarily true for every pathway. And so we have to be really careful about assuming that how we're going to get longevity is going to be, equally beneficial for like cognitive aging, which is another obsession of mine, because I really think that's probably I don't know. You can tell me. I'm obsessed with that because I feel like that's, to me, the most important aspect of our health that I'd like to keep going with age. But of course, all aspects of health are important. So, but I think that's more important than just living a long time. Now, having said that, there's some really interesting research that was done by Nir Barzilai's lab many years ago. They showed that, if you look at centenarians, so people will naturally live to be 100. I'm putting an emphasis on naturally because every centenarian out there right now has lived long, not as a result of any of the research that we're currently doing, right? So they're just people who are either mostly genetically predestined to live a long time. If you look at their health, for the most part, the way that they live longer is by delaying the onset, all these age-related diseases that most people suffer from. Cardiovascular events is a really big one. And some other aspects that come up, but like cardiovascular health is probably the biggest one. And that's why it's really exciting that, probably everyone has heard now about these, the Wegovy, so, things like semaglutide, these glucagon mimics that basically are helping people like diabetics and people lose weight. It turns out those are having very beneficial effects on cardiovascular health. So that could be, I don't think anyone developed that with longevity in mind, but it would be really surprising to me if it turns out that people who were taking these drugs and losing weight and seeing improvements in their cardiovascular function, it would be surprising to me if they didn't live longer. Mm-hmm. So that's one thing. It's kind of like, some people are directly working on longevity and some people may indirectly be finding things that really help.
Yeah. It's so much to take in.
Sorry, talk too much.
No, you do not talk too much. It's fascinating. And so, you want to think about it and then, But one of the questions that we were thinking about and have for you is that you to talk about this gut research that you're doing and how our guts play a role in longevity. I'm very interested in that.
Okay, so are you talking about things like the microbiome?
Yes.
Yeah. Okay. And to be honest, like my lab is not working on that, so I can tell you about the work that other people's labs have done. The microbiome is a fascinating field because there's been this underappreciation of the microbiome, and then to some degree maybe an overcorrection where people were attributing every good thing that could happen to microbiome health. So I think somewhere in the middle is probably the correct thing. But the idea, for example, is if you were to replace the microbiome, for example, in the intestine of a person who is old with the microbiome from a young animal, would that help them live longer? Okay. Mm-hmm. And there is some indication that might be beneficial. And so the real experimental work that I think made this true, it was true for a long time that if you move like diseased or aged microbiome into a healthy animal, this is usually done in mice, that made them sick. But that's kind of like maybe uninteresting, except for in the case where there was one study where they took the microbiome from, I believe it was mice with Parkinson's don't want to get this wrong, but a neurodegenerative disorder. And it seemed like some transfer that actually caused Parkinson's phenotypes in that mouse. So that's a fascinating area. But the other thing, if we're talking about trying to live longer and healthier, question was, can middle-aged or aged animals benefit from a young microbiome? And so that was done by Dario Valenzano's lab working in killifish. And so the idea here was that if you take that microbiome out of a young killifish and put it into a middle-aged one, would it help them live longer? And it did. And then more recently, I believe it's John Cryan's lab in Ireland, did a similar experiment in mice and showed that they had improved cognitive function. So, the microbiome probably doesn't do everything, but what young, healthy microbiome bacteria do is they secrete specific metabolites that may be beneficial to us. And so if you keep, the population of those bacteria higher and the, deleterious bacteria lower, then maybe you could have something that would be somewhat beneficial. But how that all works, especially for improving cognitive function, I think is not well understood yet. But it's, it's promising. It's one of those other things could you take something in a pill that would allow you to have healthier microbiome that would be generally beneficial? And that doesn't seem too far out of reach.
So the word itself, microbiome.
Yeah.
Yeah, we hear that a lot. But do we understand it? Well, can you just go back to the basics of that word and explain it, please?
And again, this is based on other people's research, not my own. But the microbiome is what it refers to is the collection of microbes, in particular bacteria, that are resident in our gut. And then we have lots of them that are absolutely required for our normal digestion and normal function. Mm-hmm. But if you look at animals and people with age, there's, so the microbiome in a healthy young animal is, there's a bigger concentration, a bigger proportion of these particular types of bacteria, in particular ones that kind of are associated with like things like yogurt and things. And when people age and when animals age, there's a shift in the type of bacteria that are present. Now, the tricky part with all this stuff is cause and effect. Mm-hmm. Does the bacteria shift because they are responding to a difference in our environment, or their environment, like an aged body, or are they causing aging? And I think both are probably true, but understanding which part of it we can benefit from is really important. For example, there have been ideas that the microbiome has something to do with particular disorders. But if it turns out that that disorder causes the person to eat differently and take in different foods that then support the growth of different bacteria, then it's the wrong thing to assume that the microbiome is causing the disease. Instead, it's the food changing the environment. And so that's why it's really important, because people, they'll see some headline and think, oh, I should only eat this food, when in fact it's like maybe has nothing to do with it. That's why what I tried to get to in the book is trying to really tease apart this fact from fiction and, cause and effect that we need to understand better before we start taking a lot of drugs that are based on things that may not be, based in reality.
Mm-hmm. Have you found anything that's particular to women in aging?
Okay, so the focus, I would say in particular, what we've been interested in for a long time in reproductive aging. So like all aspects of women's health, it's fascinating to realize that, and this has now changed, but when I started my lab, if you asked around, there was very little interest in understanding the question, can we predict how long a woman will be reproductive, right? But if you talk to a woman who's like 32 and has like a high-powered job, that's like on their mind all the time, right? So having a diagnostic that would tell them, well, I'll be able to have kids for another 10 years, another 5 years, or I should have already started, right? And that seems like something we should be able to figure out, but it was really hard to even get funding to ask that question at a time when, and I have to say, like, I went to a few workshops about reproductive aging. I was all excited, and then I got there and they were like, most of the talks were about men and sperm aging, and I could not understand it. Like, I couldn't understand why is this why are we throwing money at this question instead of asking, like, how long can we keep oocytes healthy? Mm-hmm. And so that led us down the road of asking, well, can we develop the little model system C. elegans as a model for this? And it turned out to be surprisingly true. You can, like, basically it turns out that in almost every animal, I think, in order to keep an oocyte healthy as it sits there in the mother waiting to be fertilized, it requires almost all the same molecules in every organism. And it's really surprising too, because of this time scales being so different. But it turns out C. elegans, they can only reproduce for about a week, but within, after about 7 or 8 days, their oocytes look terrible. And then if you look at the genes that change with age, they actually look almost identical, even to the same genes. And then we found some mutants that it can extend reproductive span and keep oocytes healthy longer. So we actually know the molecules that do that. And that led us down the road of we actually found some drugs that will, very simple things like, folic acid and other things that basically keep the oocytes functioning better with age. Now, it's a long road between like finding something like that and and getting something that will help people. Right. But at least for some of these things that are already FDA approved and very safe, we could, recommend those things. Now, for older women, menopause is actually about a 15-year gap between reproductive aging when oocytes start to decline and when menopause on average is onset. So one of the really irritating things when I first started working in this field was that reviewers would interchange these. Like they would use the words menopause and reproductive aging interchangeably and drove me crazy because in a woman's life, that's a 15-year gap, which tells you right away it can't possibly be the exact same mechanisms. So finally now, through this thing called the GCRLE, it's the Global Consortium on Reproduction and Longevity Equity, that is actually giving some money to both of those questions because they're very different and trying to understand menopause and postmenopause, all the changes that happen with aging postmenopause, that's really important to address. And so, I think there's going to be some incredible research that comes out of that area, but it's just getting, that area is just getting some funding and just getting started. Like my colleague Yusin Su at Columbia, she's been really asking this question, really focusing on menopause. So,
what's the question?
The question is like basically what happens upon menopause? Are there ways to slow down because there's greatly accelerated markers of aging. So let me just back up and say kind of terrifying study that was done a few years ago out of UCLA where the biological clock was studied. This idea of like DNA is coded with these proteins and also it has on it these methylation groups, just methylation groups that get added to the DNA that we don't really understand everything about what they do, but they seem to change with age, right? So I'm forgetting his name right now, at UCLA, Steve Horvath, he developed what's called a biological clock where he figured out that a certain set of these methylation groups in specific places on DNA actually correlated with aging, okay? So he developed this clock. And then he and Morgan Levine had a really nice paper where they asked the question, upon menopause, what happens to this biological clock? Does it still continue on the same trajectory or does it change? And their terrifying finding was that upon menopause, this aging trajectory greatly steepens. Okay, so aging is actually rapidly accelerated due to menopause. And they did some elegant, I think really nice comparisons there where they figured out that it didn't matter whether it was surgically induced menopause or, it was surgically induced. That was how they were able to, uncouple it from normal aging. And then they used hormone treatment and showed that they could slow it. So I think that does suggest that some of the problems that women see post-menopause could be addressed by some of the treatments that exist now, and then probably more that will be developed later as a result of understanding this better. Yeah, but it was really like looking at this paper, I was like, oh my God, wow, this really does, it's not anyone's imagination. There is an aging acceleration upon menopause, yeah.
What's your explanation for why women live longer than men on average?
Right. Okay, so there's a couple of things. So one thing is we have to dissociate the sociological from the medical, from the biological, right? And that's tough unless you're using a model system. Right. So in humans, we have all these things combined. For example, it seems that men, it turns out that men who have wives live longer than men who don't. Men who have a daughter who ends up taking care of him live longer than those who don't. Women who have no husband live longer than anybody else. So there's a lot of interesting things that can only be explained by social aspects and stress, and it turns out social networks are probably mostly created by women, and so men in those circles actually benefit from that as opposed to men. So and I'm sure that all that impinges on the biology as well. If we look just at the biology, though, we see there are a whole different set of factors, and that really depends on the species, which one lives longer. But for the most part, most female species live longer. Some of it could be tied to longevity mechanisms that have to be in place in order to allow them to reproduce. And so there's this really interesting connection. For example, women who, this is all going to sound backwards, but we have to look at the whole picture. So Tom Pearls did a study many years ago where he looked at historical records of centenarians and women who only lived to be, I forget, in their like 70s. And he was able to backtrack. These were like from Boston in the 1800s or something. And what he found was there a correlation between the age, women who had a child, who were able to have a child. And this is, of course, before contraception and things. So women who had kids really late, that was probably a biomarker of their future longevity. And so he found that women who were like centenarians were more likely to have had a child when they were over the age of, I think it was like 38 or 41. So the idea was that those were women who were probably always going to be centenarians, and their ability to have kids was sort of a phenotype. It's a marker of that long-lived possibility. Of course, when this got into the news, people misinterpret this right away to say, if you want to live long, have a kid late, which is not at all the story. And of course, now with artificial reproductive technologies, that kind of like, it's almost impossible to understand, like, look back and see that, but that was the finding. So, so there's a connection between being able to naturally have a kid late and long life. But I think it just reports on what's already systemically different in those women. Does that make sense?
Yeah. Did you have another question, Catherine?
Well, I was just thinking, given our audience, which is 99.9% women over, after menopause.
Yeah.
But what else would you want to highlight from your research and the questions you're asking that would be of interest to our group?
So, like I said, I'm really obsessed with cognitive aging because I think in my field, at least for a long time, there's such an obsession with longevity itself without asking, well, does the brain still work? And so we've switched focus almost like my lab is really interested in this question. And actually one of the great things is that because I became interested in this question and we developed this model in C. elegans, we were able to show quite recently that what we found in this little worm, where we figured out how to turn on one gene, hyperactivate it in this really old animal, old for worms, it was like 8 days old. Because they basically have no memory at that point. But we used the same pathway to convince our friend, Salvade at UCSF, to make the exact same mutation in the analogous protein in mice, which is exactly like what we have, and inject it into the hippocampi of 2-year-old mice. So 2-year-old mice are about average 70 to 80 year, the equivalent of 70 to 80-year-old person. And those mice actually completely recovered their memory, and they had even excellent health span. Like they were around their cages much better. It seemed like it rejuvenated them, and that part we don't understand at all. But we do understand the part for the brain where with memory, because when we got the brains back from those mice and did the molecular analyses on those, it turned on all the same genes that we'd already seen in the worms. So that pathway is extremely well conserved. And so that's given us hope that what we're finding and using worms as a screening tool to look for compounds that might help the brain could actually not in such a distant future be used for, at least we can test in mice and then test in humans. So I'm really happy about how that worked out because it was a huge gamble when I started the lab. It could have all been wrong. Like there was no guarantee that it was going to work. But luckily, by choosing carefully and really understanding the pathways that we're looking at and which ones are very well conserved in mammalian brains, we've been able to find ways that we hope that are going to help maintain brain function longer. So that's what I'm really excited about. Oh, and I should mention, because we were excited about that, and because Saul and I actually were contacted by the Simons Foundation. So Jim Simons funds a lot of excellent research. He started an autism consortium many years ago that's done a lot of great work. And so we got together and wrote a white paper, and then it became the Simons collaboration on plasticity and the aging brain. And now we're funding, I think it's something around 30 research groups to really understand cognitive aging in a way that's different from studying longevity or pathologically related dementias like Alzheimer's, because there's a lot of funding for Alzheimer's, but there's not so much just for normal aging, even though I think that's really important for all of us to keep our brains functioning.
Absolutely.
So much to take in and so much more to learn. And so we wish you a long life, Coleen, so that you can keep up this tremendous research. Really. Yes.
Yeah. Well, thank you so much, Coleen. And listeners, thank you for your loyalty. Because of you, our numbers are growing all across the country and overseas, and this is a good thing. Still, we need more subscribers and reviews on Apple Play and YouTube. So please support Women Over 70 and let your voice be heard. Help us change the conversation about women aging.